Welcome to the learning module for Oncogenesis. This is Dr. Tafke for the Cellular and Molecular Medicines course. After completing this learning module, students should be able to list the different classes of proteins involved in tumorigenesis, explain how mutations can lead to transformation, describe the difference between a proto-oncogene and an oncogene, explain why a gene is classified as a proto-oncogene or as a tumor-suppressor gene, provide examples of oncogenic transformations, explain how DNA viruses can cause transformation, describe transformation by retroviruses, explain insertional mutagenesis, and also tell how chromosomal rearrangements can contribute to transformation and provide examples. Benign tumors arise quite frequently, but usually pose very little risk as they are small and localized. As illustrated in these examples of benign tumors, they are usually well encapsulated and fairly well differentiated. Here we see the gross and microscopic views of a tumor invading normal liver tissue. On the left, the gross morphology of a human liver in which a metastatic lung tumor is growing. The white protrusions on the surface are the tumor masses. On the right is a light micrograph of a section of the tumor, showing areas of small, dark-staining tumor cells invading a region of larger, light-staining normal liver cells. You can notice the high nucleus to cytoplasm ratio with prominent nucleoli and relatively little specialized structures. It is the invasion of these cells into otherwise normal tissue that is diagnostic of a malignant tumor. Cancer is caused by mutations in somatic cells that result in transformation to a cancerous phenotype. Transformation normally requires from as few as 1 or 2 mutations to as many as 20 within a single cell. Transformation results from the action of oncogenes, which are abnormal versions of normal genes called proto-oncogenes. Proto-oncogenes are converted to oncogenes when they undergo gain-of-function mutations. The other class of genes that contribute to transformation are known as tumor-suppressor genes. They are defined by loss-of-function mutations. Most oncogenes and tumor suppressor genes are proteins that are involved in controlling cell proliferation. These can include growth factors, growth factor receptors, intracellular effector regions of growth factor receptors, signal transduction molecules, especially protein tyrosine kinases, second messenger proteins, intracellular receptors, transcription factors, proteins that regulate cell cycle control or checkpoint control proteins, as well as proteins that regulate DNA repair and also program cell death. There are several mechanisms by which a proto-oncogene can be converted to an oncogene. A point mutation can result in an abnormal, hyperactive signal transduction protein. Gene duplication and gene amplification events can result in overexpression of a normal protein involved in activating cell proliferation. Chromosomal translocation events can result in the expression of higher than normal levels of a normal protein involved in proliferation, or in the expression of an abnormal hyperactive protein. Localized DNA rearrangements, creating insertions and deletions or inversions or transpositions, can result in hyperactive abnormal proteins that drive proliferation. And insertion of viral DNA sequences can drive the expression or overexpression of normal proteins, creating hyperproliferation signals. All of these events are gain-of-function mutations. Gain-of-function mutations act dominantly. Only one allele of a gene needs to be mutated in order for the phenotype to appear in the cell. Gain-of-function mutations can occur in growth factor receptors that regulate cell proliferation. For example, a member of the epidermal growth factor receptor superfamily known as the new receptor because it was first identified in a neuroblastoma, is a HER2 receptor bearing a point mutation in the plasma membrane spanning domain in which a valine has been converted to a glutamine residue. The new receptors dimerize and autophosphorylate in the absence of ligand binding. Another example is deletion of the growth factor binding domain of the epidermal growth factor receptor HER1, also known as ERBB. Deletion of the extracellular growth factor binding domain allows the truncated receptors to dimerize and autophosphorylate in the absence of epidermal growth factor binding. This creates a hyperproliferation signal in the absence of growth factor stimulation. Constitutively active signal transduction proteins are encoded by many oncogenes. For example, the Rous proto-oncogene becomes an oncogene when mutations reduce its GTPase activity. Rous mutations are common in many forms of cancer. Loss of GTPase activity in Rous means Rous remains active longer than required. Proteins that function as guanine nucleotide exchange factors to activate Rous are also proto-oncogenes. If they become hyperactive, Rous activity is accelerated in the absence of appropriate signaling. Conversely, proteins that function to switch Rous off, the GTPase activating proteins such as neurofibromin or NF1, function as tumor-suppressor genes because they switch Rous signaling off. Proteins that control the checkpoints in the cell cycle can also contribute to transformation. Mutations resulting in overexpression or hyperactivity of cyclin and cyclin-dependent kinases are oncogenic, while mutations that inactivate retinoblastoma or cyclin-dependent kinase inhibitor functions result in inactivation of tumor suppression. Approximately 15% of all cancers are due to cancer-causing viruses. There are two types of viruses, DNA viruses and RNA viruses. Of the DNA viruses, papillomaviruses, hepatitis B virus, and the Epstein-Barr virus are all known to contribute to human cancers. Of the RNA viruses, the human T-cell leukemia virus and the HIV virus are also known to lead to some forms of cancer. DNA viruses normally do not integrate into the genome of the host cell, and the oncogenes in DNA viruses are integral parts of the viral genome that are required for viral replication. These proteins are utilized by the virus to take control of the host cell replication machinery, but usually this is a well-balanced and well-controlled event that leads to only a small amount of proliferation of infected cells. The result normally would be a benign growth, such as a wart. However, occasionally this process goes awry, resulting in a malignant cell type. RNA viruses, on the other hand, integrate their genome into the host cell's genome. The oncogenes in these transforming retroviruses are not part of the normal viral genome. They are not required for viral replication. These sequences are extraviral DNA information that, when expressed, cause transformation of the infected host cell. This figure illustrates how a DNA virus, such as a papillomavirus, can cause a malignant transformation. Normally, DNA viruses do not incorporate their genome into the genome of the host cell. The virus uses host cell machinery to transcribe and translate viral genes to produce viral proteins which interact with cell regulatory proteins and cause the cell to take on a hyperproliferative state, thus ensuring that there are adequate supplies available for production of new viral particles. The result is a hyperproliferative state which only lasts for a period of time. and usually results in a benign growth or wart. However, occasionally, the viral DNA or a fragment of the viral DNA can accidentally integrate into the host cell's genome. If this viral DNA fragment happens to be a gene for one of the cell cycle regulatory viral proteins, its expression can become unregulated and unbalanced, resulting in transformation of that cell, which will then hyperproliferate and collect mutations, which will further it along the path to becoming a malignant tumor. An excellent example of a DNA virus control protein is the SV40 large T protein. Two important cell cycle regulatory proteins are the retinoblastoma protein and the p53 protein. Both proteins function to halt cell proliferation. Retinoblastoma normally binds to and sequesters essential transcription factors called E2F that are necessary for the S phase of the cell cycle, while the function of p53 is to halt the cell cycle when conditions aren't correct, such as the presence of damaged DNA. SV40 is a transforming DNA polyomavirus. It makes two early proteins called large T and small t by an alternative splicing mechanism. Large T binds to and inhibits both retinoblastoma protein and p53, thus subverting the host cell's cell regulatory mechanisms. Expression of large T is sufficient to transform many primary cells in culture. The human papillomaviruses, another DNA virus, express two separate proteins, E6 and E7, that function like the large T protein of SV40. HPV E6 binds to p53 and inactivates its function, while HPV E7 binds to retinoblastoma protein and inactivates it. There are two basic mechanisms by which RNA viruses, or retroviruses, can cause transformation. Retroviruses normally integrate their genomes into the host cell and then use the cellular transcription machinery to replicate the viral genome. If the virus integrates at the site adjacent to a proto-oncogene, occasionally read-through transcription will transcribe part of that proto-oncogene along with the viral genes. This extra viral piece of DNA containing part of a proto-oncogene is packaged into a viral particle. When this viral particle infects another cell, the extra viral piece of DNA can be transcribed into a functional protein, resulting in the expression of an oncoprotein and transformation of the cell. The genome of a normal RNA or retrovirus, which will not cause transformation, looks essentially like this. It has two long terminal repeat sequences which are used to guide integration into the host cell genome and regulate expression of the viral genes, which are in between the LTR sequences. Normally, these retroviruses have three genes, a GAG, POL, and envelope gene. The GAG proteins are nucleocapsid proteins. The POL gene encodes an RNA-dependent DNA polymerase or reverse transcriptase, and the envelope proteins necessary for production of new viral particles. All three of these genes are required for normal retrovirus replication. In comparison to normal retroviruses or non-transforming retroviruses, analysis of the genome of a transforming retrovirus, such as the Rous sarcoma retrovirus, revealed that there are extracellular, non-viral DNA sequences present in the genome. For example, the Rous sarcoma retrovirus, which causes sarcomas in chickens, was found to contain extraviral DNA sequences which were termed V-sarc, for viral sarc. The V-Sarc sequences were found to be required for transformation, but not for viral function. Normal cells contain a gene that is very similar to V-Sarc called C-Sarc, which is a tyrosine kinase, similar to the JAK kinases involved in cytokine signaling. The C designation indicates the cellular form of Sarc. C-Sarc is a proto-oncogene, while the viral Sarc is an oncogene. It is a protein tyrosine kinase that is constitutively active. The cellular Sarc protein is a tyrosine kinase whose activity is modulated by phosphorylation of a critical tyrosine residue located in its carboxy terminus. When this tyrosine is phosphorylated, Sarc is switched off. V-Sarc, or viral Sarc protein, often has a deletion of the carboxy terminus. This deletion removes that critical tyrosine residue, resulting in a Sarc tyrosine kinase that cannot be switched off. Examining the genomes of transforming retroviruses led to the discovery of many signal transduction proteins we've already discussed in cellular and molecular medicine. Basically, the transforming retroviruses cloned these signal transduction proteins for us before we understood anything about their role in signal transduction pathways. These proteins include the RAS family, the RAF MAP kinase family, and the epidermal growth factor, or ERBB, receptor tyrosine kinase families. The other mechanism by which RNA viruses or retroviruses can cause transformation was discovered by studying the so-called slow-acting carcinogenic retroviruses. Unlike the transforming retroviruses, the genomes of these slow-acting carcinogenic retroviruses lack extraviral DNA sequences in their genomes. Infection with a slow-acting carcinogenic retrovirus does not always lead to transformation. They only cause transformation when occasionally they insert and disrupt a tumor-suppressor gene or near a proto-oncogene such that the long terminal repeat sequences can activate expression of that nearby proto-oncogene. Thus, whether or not the retrovirus is transforming is determined by where the viral genome inserts into the host genome. Thus, it is referred to as insertional mutation. For example, insertion of a retroviral sequence near the MYC proto-oncogene can result in overexpression of the MYC transcription factor, whose overactivity promotes transformation. Alternatively, if the retrovirus inserts into the gene encoding a tumor suppressor, such as the retinoblastoma gene, expression of the RB gene can become disrupted, and the cell will lose some tumor suppressor capability. Because the insertion site of the retrovirus genome is random, it's just bad luck when it inserts next to a proto-oncogene or inside of a tumor suppressor gene. An example of a slow-acting carcinogenic retrovirus is the mouse mammary tumor virus, or MMTV. Examination of mammary tumors in mice revealed that integration of MMTV genome next to a proto-oncogene called Wnt1 leads to overexpression of the Wnt1 protein, which provided a growth advantage to those cells. Analysis of numerous mouse mammary tumors created from MMTV-infected mice revealed that insertion anywhere within the vicinity of about 10,000 nucleotides from the Wnt1 gene resulted in transformation. Chromosomal translocations can also result in transformation. A translocation can cause elevated expression of a proto-oncogene by placing the proto-oncogene under control of a different gene promoter. This is what happens in Burkitt's lymphoma, which is a B lymphocytic tumor. It's caused by a translocation between chromosome 8 and chromosome 14, in which the proto-oncogene c-MYC is placed under the control of an immunoglobulin promoter. Since immunoglobulin promoters are highly active in B cells, this results in overexpression of the c-MYC transcription factor, which then drives replication of these cells. Chromosomal translocations can also create a fusion protein, which has aberrant and unregulated activity. A reciprocal translocation between chromosome 9 and chromosome 21 will generate what is known as the Philadelphia chromosome. This translocation creates a fusion protein between a tyrosine kinase known as the ABL tyrosine kinase and a region on chromosome 22 known as the BCR, or breakpoint cluster region. This creates a fusion protein BCR-ABL that has constitutive tyrosine kinase activity, which drives excessive proliferation of a clone of stem cells. These stem cells then differentiate into adult, well-differentiated white blood cells. A subsequent mutation, for example in the p53 gene, results in an acute leukemia that was normally fatal within 5 years. The BCR N-terminal segment from the Philadelphia chromosome BCR-ABL fusion gene forms a coiled coil domain. ABL is a tyrosine kinase which is normally localized to the nucleus. The BCR-ABL oncoprotein, however, is a cytosolic tetramer that is constitutively active as a tyrosine kinase. It acts on proteins that are not normally substrates of the ABL tyrosine kinase, leading to their activation in the absence of growth factor stimulation. These proteins include the JAK tyrosine kinases, STAT transcription factors, and PI3 kinase. Imatinib, or Gleevec, is an inhibitor of the BCR-ABL tyrosine kinase. It is the first example of targeted chemotherapy to inhibit an oncoprotein. Gleevec binds to the ATP binding site of the ABL tyrosine kinase, preventing phosphorylation of substrate proteins. Prior to the development of Gleevec, CML was fatal for 98% of patients within 5 years. Now, most patients on Gleevec live normal lives by taking a daily oral medication. Here is a short quiz to test your knowledge of transformation mechanisms.